Good day, everyone. I'm here to present our paper entitled Preliminary Identification of Flacidiplodia pseudotheobrome Causing Tomato Fruit Rot in the Philippines. In our country, tomatoes are the most profitable crops and rank third as the most important vegetable in terms of volume. However, several fungal species cause diseases on both pre-harvest and post-harvest tomato. One of the many pathogenic fungi includes the Lashiodyplodia species, which is characterized by mature and dark brown conidia with thick walls and longitudinal striations. For our objectives, this study includes the isolation of Lashiodyplodia species from tomatoes with symptoms of rot, identification of Lashiodyplodia isolates by morphology and by ITS gene barcoding, and confirm the pathogenicity of the isolates obtained. For methods, tissue samples were collected at the tomato farm in Alaminos, Pangasinan. The causative fungus was isolated from infected tissues using the single spore method and cultured to PDA for morphology identification. Meanwhile, the microscopic morphology was observed in the 7-day culture of the isolate. The isolate then undergo pathogenicity test and phylogenetic analysis, which was done using the ITS barcode and compared to the reference sequences of Flaciodiplodia species from published literature. Lastly, using the MegaX application, multiple sequence alignment was done as well as the construction of phylogenetic tree. For the result, the 7th-day colony of the isolate is fast-growing and appeared grayish-white, woolly with abundant aerial mycelium. The reverse has pale yellow to off-white color. The conidial genus cells are pale brown, smooth, opifiform, and young conidia are hyaline, unicellular or septate, oblong to ellipsoid, and have thin walls, while mature conidia are dark brown, have one septate, ellipsoidal, thick-walled, and with visible longitudinal striations. The phylogenetic tree showed that the data from the ITS sequence of the isolate revealed that it is closely related to other Lashiodyplodia species, particularly with Lashiodyplodia pseudotheobrome with a bootstrap value of 82%. For pathogenicity test, the lesion is spread to the whole tomato fruit within 3 to 7 days, resulting in the sogginess of the fruit. In conclusion, the isolate was identified as Lashiodyplodia pseudotheobrome based on the morphological characteristics and phylogenetic analysis using the ITS gene sequence, and it was responsible for causing diseases on the tomato fruit. However, the phylogenetic tree from ITS gene sequence was insufficient for the identification of the species because of the low bootstrap values. Therefore, final identification of the isolate requires a multiple loci sequence analysis which includes the ITS, BT2, and TEF1 alpha genes. And that concludes our presentation. Thank you for listening.